Hello students, thank you for watching my lecture Synthesis of Proteins. In the previous lecture we have seen how solid phase synthesis can be used to covalently connect different amino acid building blocks together to form peptides. The size limitations there is around 50 amino acids, peptide fragments in length. Today I would like to show you how we can produce larger peptide units or to synthesize even whole proteins by assembling different peptide fragments together and connecting them finally with native peptide bonds. The principle of peptide fragment condensation is very simple. You start out synthesizing the peptide fragments that cover the whole sequence of the proteins, and in the final step, you just put them together and connect them by native peptide bonds. However, with classical chemical methods, this is not possible, and that's why two strategies have been developed in the past to achieve the goal of fragment condensation. The first one is native chemical ligation, and the second one is the utilization of peptide ligases. Let's start with the development, how native chemical ligation was developed. Before native chemical ligation technology was discovered, researchers tried to couple large peptide fragments together to form then functional proteins. However, they always used a coupling strategy that involved the formation of a non-native peptide bond, which at that time was the limit of the technology. Here you see one of these examples. This is the synthesis of an analog of HIV-1 protease. This work was done in 1992. What was introduced here is on the peptide fragment covering the amino acids 53 to 99, there was a bromoacetic acid derivative coupled to the N-terminus, and at the C-terminus there was a thiocarboxylic acid introduced, and these two complementary functionalities, they can react in a nucleophilic substitution reaction to form a covalent connection, but important to note, not a real peptide bond. So a covalent connection was established between the two fragments, and via this fully active HIV-1 protease was chemically synthesized. So let's now move to the real native chemical ligation. And native chemical ligation rests upon a finding that is really old, and this is you see here a publication still in German that describes the coupling of two amino acids. On the one hand, this is cysteine that is coupled to valine. The valine had been functionalized as a thioester, and it was stated in this early publication in 1953 that a peptide bond could establish between the two amino acids. And then for a long time, this research achievement was forgotten until the day when Steve Kent published a famous paper in Science in 1994, where he established the first chemical synthesis of a peptide using two fragments, and he used this early discovery of coupling two amino acids. He used this to not only couple two amino acids, but to fuse two peptide segments together that had been synthesized by solid phase synthesis. So in the first peptide fragment, there is a thiobenzyl ester introduced at the C-terminus. The fragment 2 contains N-terminal cysteine, and what happens during this fragment concentration is the cysteine attacks the thioester, and then a transesterification occurs, and in the next step, then a spontaneous SN acyl transfer reaction takes place, which is irreversible and which yields to the formation of a native peptide bond between the two fragments. And in this fashion, it was possible to synthesize a fully functional interleucine 8 that is not distinguishable from the native protein. I would like to give you another example that was almost published 10 years later from the first Native Chemical Ligation paper. In this case, the for sure the target of synthesis is more complex than the previous one, and in this case the goal was to synthesize an erythropoietin, or short EPO, that was modified, and this synthetic EPO should be investigated in how the protein could be improved by introducing synthetic moieties. Therefore, I need to tell you a little bit more about the protein. EPO is a glycoprotein consisting of 165 amino acids, and the protein part is only 40%, while the sugar part that is attached to the EPO backbone represents roughly 40% of the molecular weight. EPO, all the sport interested students might know this very well, especially when watching cycling. EPO is a very famous doping reagent because it helps to stimulate the formation of red blood cells. However, it's not only good for sportsmen to unethically improve your record, but it is also a protein drug that was used quite frequently in for different indications, as you can read on the slide. And at least in the year 1995, it had a quite huge yearly turnover of $2.5 billion. So it was a protein that was established on the market. And the research question that was posed here was when the glycosylated parts, so when the sugar parts are replaced by polymer units, polyethylene glycol, branch polyethylene glycol units, what this synthetic modification or how these modifications on the protein backbone, how these modifications relate to different properties of the proteins, and especially the question was asked whether by such polymer modifications the properties of this protein could be improved. So here on the right-hand side, you see now the synthetic pathway towards the synthetic erythropoietin, so SEP. Altogether, it is made from 4 different peptide segments, shown here in different colors. What you also recognize on this synthetic scheme is that 2 of the fragments contain the branched polyethylene glycol modification. This is the first segment and the last segment. All the peptide fragments were synthesized by the Merrifield approach, so using Bock protection group strategy. The yields of the peptide synthesis of the individual fragments were 15 to 30%. The yields for coupling the polymer functionalities to the peptide units via oxime formation were 30 to 50%, and the yields for native chemical ligation varied between 40 and 70%. And finally, to do all the tests, then 100 milligrams of the material were synthesized. So this material was really done in large scale. And important to mention here, the peptide couplings were the only way, the native chemical ligation was the only way to achieve such a synthesis of the protein. So that was the major breakthrough regarding this work. For sure, the material needed to be characterized, and you see a SDS page trail on the bottom left. You see a AC mass spectrum, and you also see a Multitoph mass spectrum indicating on the right hand side, indicating the synthesis of the right protein. So now the material needed to be biologically evaluated, and for sure it was compared to the native EPO. The stimulation of, or the cell proliferation of UT7 cells between EPO and SEP was more or less the same. So the UT7 cells that are bone marrow cells that are directly stimulated by EPO. So both proteins, the native one and the synthetic one, showed similar stimulation activity. When it comes to the hematocrit value, that's the volume fraction of red blood cells in the whole blood in a rat model, showed that the synthetic counterpart was more effective than the native proteins to drive up the hematocrit value. And finally, the stability tests showed that once you replace the sugar units in the native EPO by synthetic polyethylene glycol units, the stability of the protein in red blood was increased, which probably leads to the biological effect of the higher hematocrit value that you can see on the middle scheme. So let's move now from the native chemical ligation method to the peptide ligases. Peptide ligases use a different principle than native chemical ligation. In this case, protein scaffold of a protease is used to assemble two peptide fragments together. And the principle works like that you take a protease scaffold, you mutate the protease activity by inserting mutations, and one of the examples that is known is the formation of aspartame, which is artificial sugar, or a sugar with less calorie than the carbohydrates. This is an industrial example where peptide ligases are commercially used. Today I would like to discuss with you an example that's called subtiligase. And subtiligase originates from or is derived from the protease that had been engineered. In this case, there's a protease called subtilisine, and in subtilisine, in the active site, a serine was replaced by a cysteine, and a proline was replaced by an alanine. And with this mutations, the protease activity was, so to say, abolished or diminished, and in this way, the ligation function could be improved. So what we do here is, in principle, we do a reverse proteolysis. Therefore, the peptide segment at the C-terminus needs to be functionalized as a peptide ester, as you can see here. So here is a glycine phenylalanine peptide ester introduced at the C-terminus of the peptide that should be ligated. The glycine phenylalanine acts as a leaving group, and when you now use subtiligase, a second peptide with an conventional N-terminus can be ligated to the first peptide fragment. In this case, RNase A was built up from 5 fragments. Altogether, the protein contains 124 amino acids, and on average, the coupling yield per step is around 75%, which is a quite remarkable value. Again, when looking at this paper, you can see that a lot of non-natural building blocks and polymer modifications have been introduced in the proteins using the engineered protease, and all the building blocks to make the final protein were again synthesized by solid phase peptide synthesis, allowing the introduction of these modifications. If it now comes to the synthesis of even larger proteins, or you don't really need too much synthetic modifications in your protein, then you can use classical recombinant DNA technology to fabricate your protein or peptide of choice. I know that this is the topic of probably different lectures during your lecture series in the Max Planck School. However, I would like to briefly give you as an appetizer and one slide overview how this works. What is usually done is you take a plasmid onto this plasmid, which is circular double-stranded DNA, with the help of restriction enzymes, and again with the help of DNA synthesis that is also done on solid phase, and the formation of larger nucleic acid fragments. You can design your gene of choice, and then you can ligate it into this double-stranded circular piece of DNA. Once you have cloned your gene inside the plasmid. You take the plasmid, you implant it back into bacteria, and then you can grow the bacteria, and at one point you induce a stimulus, and with this stimulus the proteins start to read the DNA and express the protein of choice that you have inserted or that you have encoded on the plasmid. Once the protein synthesis in the bacteria has been done, you disrupt the cell wall of the bacteria, and then you purify your overexpressed protein of choice. And via this method, it's also possible to make quite large quantities of proteins using recombinant DNA technology. And with this now, you have a quite good overview about the possibilities how you can make modified or native proteins using very different approaches. With this, I'm at the end of my lecture, and I would like to thank you very much for your attention.